In this video, we can discuss about the evaluation of disinfectants. As we all know, disinfectants are the chemical agent which are used for the disinfection. And the disinfection action may be either by killing the microorganism, that means bactericidal action, or by inhibiting the growth of microorganism, that means bacteriostatic action. So the disinfectant can be evaluated for their bacteriostatic effect or for bactericidal effect. And bacteriostatic evaluation can be done by serial dilution in fluid media, cup plate, fish spine bead, cylinder or filter paper method, gradient plate method or ditch plate method. And bactericidal effects can be studied by using end point or extinction time method, counting method or turbidimetric stent method. Now coming to the bacteriostatic test. These tests are to evaluate the ability of disinfectant to prevent the growth of microorganism. Now we can perform the bacteriostatic test for liquid disinfectant by serial dilution in fluid media, serial dilution in solid media, cup plate, fish spine bead, filter paper method, gradient plate method, ditch plate method. And we can perform semi-solid disinfectant by cup plate method and ditch plate method, and solid disinfectant by inhibition of seeded agar method. Coming to the first method, serial dilution in fluid media. In this, graded solutions of test substances in a nutrient media is inoculated with the test organism and then it is incubated. So from this we can observe a concentration. From that onwards it will prevent the detectable growth in the nutrient media. So that concentration can be measured as the bacteriostatic activity. So this is the minimum concentration of disinfectant which will prevent the detectable growth, and this concentration is known as minimum inhibitory concentration, and this can be taken as a measure of bacteriostatic activity. So that is the serial dilution in fluid media. Now coming to serial dilution in solid media. This method is used for the turbid bacteriostatic solution or solution which will give turbidity with fluid nutrient media. So it will be difficult to distinguish the turbidity due to the growth of microorganism or turbidity due to the bacteriostatic solution. Now in this method, nutrient agar is diluted with equal volume of bacteriostatic solution and then it is poured into an empty sterile petri dish. But in case of solid media, we have to make sure that correct inoculum density should be there. A separate petri dish should be used for each concentration of disinfectant. After diluting the nutrient agar with the equal volume of bacteriostatic solution and then pouring into the sterile petri dish, it will be dried by incubating at 37 degrees Celsius for 1 hour with lid slightly raised. Then a drop of 24-hour broth culture of test organism are placed on the dried surface and incubated for 2 to 3 days. From this, the minimum concentration of disinfectant which will prevent the detectable growth is taken as a measure of bacteriostatic activity, and this concentration is known as minimum inhibitory concentration. So if we are diluting the nutrient agar, that is the solid media, in liquid form with the graded concentrations of disinfectant, and we will pour this into sterile empty petri dish. After solidifying, the surface is inoculated with the 24-hour culture of testing sample. Then we will incubate to 2 to 3 days. So we will get a concentration in which it will prevent the growth of microorganism. So lowest concentration of disinfectant which will inhibit the growth of microorganism is known as minimum inhibitory concentration, and which is taken as a measure of their bacteriostatic activity. Now coming to the next method, coupled fish spine bead, cylinder and filter paper method. These methods are similar to the method which are used for antibiotic assay. The nutrient agar is melted and cooled and inoculated with the test organism, and then it is poured into the sterile empty petri dish. In cup plate method, when the inoculated agar is solidified, a hole about 8 mm diameter is cut in the medium with cork borer. Then the disinfectant is placed into these holes. In all other three methods, the antimicrobial agent are placed either in fish spine bead or by using a cylinder or disc, or by dipping in a filter paper is placed on the surface of solid agar. So this is an example for coupled method. In this, a bore is made by using borer, and the disinfectant solution is poured into this hole. And other method, we will place the fish spine bead or cylinder or disc or filter paper on the surface of this solidified nutrient agar which is inoculated with the test microorganism. So after the incubation, if we are getting a zone of diameter, it will indicate the inhibition of growth around that area, and the diameter of zone of inhibition is determined, and it will give an indication of relative activity of antimicrobial substance against the test. But this test can give a false indication because the development of zone of inhibition is mainly depends on the ability of antimicrobial agents to diffuse through the agar. So the antimicrobial agents which have an capacity to diffuse more to the agar will show a wide zone of inhibition, and which are not able to diffuse will show a small zone of inhibition. So it may give an false indication. Now coming to the next method of bacteriostatic evaluation, that is gradient plate method. In this, two layers of agar are prepared to form two wedges A and B. Wedge A have a nutrient agar with antimicrobial agent, and wedge B a nutrient agar without antimicrobial agent. So after the overnight incubation, the diffusion occur with the maximum concentration of disinfectant in the wedge A at the area with greater thickness of the agar, and solidified agar is streaked with the specified organism along the concentration gradient, and then it is incubated. So this is a grade plate method. First we will prepare a wedge with only agar, then another wedge with agar with nutrient agar. Then we will inoculate this by streaking method. So after the incubation, the microorganism will grow where the concentration of disinfectant is below the critical level. So from this, the minimum inhibitory concentration can be calculated by an equation MIC is equal to C into X by Y, where C is the concentration in milligram per ml of the total volume, X is the length of growth in the centimeter, and Y is the total length of possible growth in centimeter. Now coming to the next method of bacteriostatic evaluation, that is the ditch plate method. In a nutrient agar plate, a trough or ditch is cut out and a solution of antimicrobial substance is introduced to ditch to fill 3/4 of it. A loop full of each organism is streaked outward from the ditch on the surface. After the incubation, we can see the organism which are resistant to antimicrobial will grow right up to the ditch, and susceptible organism will show a zone of inhibition adjacent to the ditch, and the width of inhibition zone is an indication of antimicrobial activity of the substance. So this is an picture of ditch plate method. Here or ditch or trough will be made, and we will fill the disinfectant solution up to 3/4 of this ditch, and then we will streak the microorganism with the test microorganism. So from this we can observe the organism which will resistant will show growth entire surface of the ditch plate, and which are susceptible will show a zone of inhibition, and the width of zone of inhibition indicate their bacteriostatic activity. So that is the ditch plate method. So that is all about the bacteriostatic evaluation of disinfectant. Here we will only evaluate the capacity of a disinfectant to inhibit the growth of microorganism, not the ability of killing of microorganism.